This video is going to cover B lymphocytes, which is another type of lymphocyte involved in specific defense, which is part of the immunology part of Higher Human Unit 3. So B lymphocytes are specific white blood cells, very much the same as T lymphocytes are, okay? But they carry out a different function. So T lymphocytes carry out apoptosis, okay? Whereas B lymphocytes will produce specific antibodies, okay? So you'll probably remember that from National 5, okay, that lymphocytes produce antibodies. Now, for this, it's far more specific to B lymphocytes now. So B lymphocytes produce antibodies. Now, at this point, you can never get bored of saying the word specific, okay? Now, antibodies are Y-shaped proteins, okay? So think about proteins and the fact that these cells would then have a lot of ribosomes and would need a lot of energy to then produce those. Now, antibodies have a particular area on them called a receptor binding site. Now, that receptor binding site is specific to an antigen, okay, on a particular pathogen. So these are specific to an antigen on a particular pathogen. That's a really common phrase to use, okay? So if you'd said in a sentence in an essay question, B lymphocytes produce specific antibodies in response to a specific antigen on a pathogen, you would get quite a number of marks for that. Okay, so to describe the receptor binding site, just as a quick diagram, this is a super zoomed in version of a poorly drawn antibody. Okay, so it's Y-shaped roughly. And this area here is the receptor binding site. Then notice it's got a particular shape. So different antibodies will have different shaped receptor binding sites because it will match a specific antigen on a particular pathogen. So what I'm going to do is run through this diagram here that I've got and label key parts, and then I'm going to talk you through the step-by-step parts of what happens to this. Okay, so this here is a pathogen, and this is the antigen on that pathogen. Okay, these are all the same pathogen. Right, now notice the antigen is a particular shape, so that's that foreign protein on that foreign molecule. Now this here is a B lymphocyte. Okay, and you'll notice they have receptors on them. Remember, these receptors are made of proteins. Now, just like any lymphocyte, regardless of which type, these lymphocytes will bind to that foreign pathogen. Okay? So that that antigen will bind to that receptor on that B lymphocyte. Now, when any lymphocyte, regardless of type, binds to a foreign antigen, what it will do is it will produce clones. So remember, it will divide and it will produce clones. Alright, now as well as that, because these B lymphocytes produce specific antibodies, the cells that are, these B lymphocytes, the cells that are involved in this will have to produce those antibodies. So these Y-shaped proteins, in a very zoomed out way, are antibodies. Okay, so these clones are produced, but also the cell starts to produce antibodies. Now those antibodies then bind to those antigens on that foreign pathogen. So when they bind to them, this here, where the antigen and the antibody bind, is called the antigen-antibody complex. Okay, so all that means is basically where the antigen and the antibody bind together. Now, that binding of the antibody to that foreign antigen renders it really susceptible to getting destroyed by other white blood cells, okay? And in we come with our kind of cleanup crew again. This phagocyte will then engulf and digest and therefore carry out phagocytosis on that antigen-antibody complex, okay, of antigens and antibodies bound together, and therefore that foreign pathogen will get destroyed. Okay, so T lymphocytes carry out apoptosis, B lymphocytes carry out the production of antibodies, and therefore that antibody-antigen complex gets broken down by phagocytes. Okay, so description then of how this works. So B lymphocytes attach to an antigen on a foreign pathogen. Right? That binding then triggers B lymphocytes to do a couple of things. So it produces clones, okay, as always, and it produces specific antibodies. Right? So these antibodies then bind to antigens on that foreign pathogen, and then they inactivate them. So they basically render them useless. They stop them from being able to work. And they form an antigen-antibody complex. So that's where the antigen and the antibody bind together. Now that complex, when they've got the antigens and the antibody combined, then is destroyed by phagocytes. Now, again, as a reminder, B lymphocytes have large numbers of ribosomes to produce those antibodies because these antibodies are proteins, right? And just the same as they would have a large number of mitochondria to produce energy to be able to do that, okay? And then they would have DNA or genes in the nucleus that would then control or code for the production of those antibody proteins. Now, the last part about B lymphocytes is similar to what we talked about the last part of T lymphocytes. So these things can go wrong, okay? They can stop working. And when B lymphocytes stop working, they can become hypersensitive to normally harmless antigens. Okay, so the whole point of having B lymphocytes is to respond to harmful antigens, like viruses, like bacteria, things like that. But sometimes those antigens are harmless, right? Like pollen, like the antigens on dog hair, or like the antigens in dust. So when that happens, this is known as allergy. So B lymphocytes are responsible for allergies if they are overactive and hypersensitive. Okay, so T lymphocytes are involved in autoimmune disease, B lymphocytes are involved in allergy. Right, so that sums up the B lymphocytes part of it, and I'll move on to the secondary and the primary immune response in the next video.